Okay, good morning, everyone. So we are going to discuss our new topic. This will be about carbohydrates. Okay. Okay, so you see here the structure of glucose. This is the straight chain form of glucose. This is one of the most important monosaccharides, since this is the main molecule we use we convert to energy, to ATP, in the cytoplasm and the mitochondria. So through the process of glycolysis and through the Krebs cycle and eventually the electron transport chain, which is basically overall it's the process of cellular respiration, where the glucose molecule is converted into energy. So this is one of the most important carbohydrates we need to study. So at the very least, you need to remember its structure. Okay. In solution, it will form a cyclic structure like this one. And these are all conformations of your glucose. Okay. So as an introduction, carbohydrates are polyhydroxy carbonyl compounds, meaning you have the C double bond O. So that's the carbonyl group. So either you have an H attached to it or it's the bridge between two carbons. Okay. So their derivatives are also considered carbohydrates. And if these structures combine, you form polymers. So these are all, the polymers are also considered carbohydrates. So the simplest carbohydrates possible are glyceraldehyde and dihydroxyacetone. So these are the simplest monosaccharides possible. So we can classify carbohydrates according to the number of units in the polymer. So if it's just a single unit, if it's just a monomer, we call it a monosaccharide. So it cannot be hydrolyzed into simple sugar. So example is glucose, which I have shown you earlier. If you combine two monosaccharides, then that will be a disaccharide. So your molecules that if you have a monosaccharide or disaccharide, they usually end with OSE. Higher than that, you have oligosaccharides, so 3 to 10 monosaccharides. Maltotriose is an example. And then if you have glucose units combined, if you have more than 10 glucose units, actually it's more than 1,000, you have starch in plants, and you have also glycogen. So oligosaccharides are condensation products of 3 to 10 monosaccharides, while polysaccharides you have more than 10 monosaccharides. Examples are starch. So these are basically units of glucose combined with one another. So you can find this in plants. In animals and in humans, we have glycogen. So these are all glucose units that we can break down in order to get the glucose which we need for cellular respiration. So we go to monosaccharides. How are they classified? So there are three categories. So if you want to find the number of atoms, you use the prefix. If it's classified according to the number of atoms, the prefix tri means 3, tetro means 4, pento means 5, hexo means 6, 7 means uh, hep means 7. So it's like the your polygons, the prefixes that count the number of sides. So it's easy to remember. Okay, so that's one way to categorize monosaccharides. And then in terms of functional group, either you have a carbonyl group or either you have an aldehyde or a ketone. So if it's found in the edge, of course the aldehyde is found in the edge, you call it aldose. If the carbonyl group is found in the middle of two carbons, then it's a ketone, so you call it a ketose. The third category is you can use both. You can combine the number of carbon atoms and the uh, type of carbonyl group. So if you have a three carbon monosaccharide with an aldehyde on it, then it's an aldotriose. If you have a ketone group in a monosaccharide that's made up of three carbons, then it's called a ketotriose. So this example, glyceraldehyde is a aldotriose, while the one on the right side is an example of a ketotriose. Okay, so using the third category, how will you classify this type of monosaccharide? This one at the bottom. Anyone? Anyone who wants to recite? What do you call, so the third category where you combine the number of carbons and the type of carbonyl? What do you call this monosaccharide? Yes, Miss Aguila.
Sir, try lang.
It what?
Ket- Ketros.
Yes, it's a tetros. Okay, so from the first category, it's a tetros. So what functional group do you have there? Yes, Miss Kasidsid.
Sir, try lang po. Is it aldose tetrosis po?
So it's an aldose and it's also a tetros. So you can combine the two names. It's an aldo tetros. Okay, so for the first category, the number of atoms, that's correct. It's a tetros. For the second, what functional group you have, it's an aldehyde, so it's an aldose. So if you both combine the number of carbon atoms and the carbonyl group, then this is an aldo tetros. Okay. Okay, so this is a monosaccharide. It's basically a carbohydrate that cannot be split or hydrolyzed into smaller carbohydrates. So it's the monomer. It's a basic unit of the carbohydrate. So you have 3 to 9 carbons long, and you have a carbonyl group. So it's either an aldehyde or a ketone. You have several hydroxyl groups like this one. So there's always an OH attached to carbon, as long as it's not the carbonyl group. So this is the one we talked about, aldotetrose. So the name of this structure is erythrose. Okay, so because the carbonyl is found here, C double bond O, and then you have H, it's an aldose. And according to the number of carbons, it's a tetrose. So it's an aldotetrose. So this is another important structure in monosaccharide. This one is a hexose. So it's a ketohexose. So we call it a fruit sugar. We can find it in fruits. Combined with glucose, it's what makes up our common table sugar. So the disaccharide formed by this fructose and glucose is the common table sugar. So there's a ketone group on the second carbon. So everyone else, every carbon has an OH group attached to it. You saw the first structure earlier. So the simplest aldose is glyceraldehyde, and then the simplest ketose is dihydroxyketone. So you have the same molecular formula, therefore they are constitutional isomers by the fact that the groups are very different. The the only similarity between the two molecules are basically its molecular formula. You see here it's a ketone, this one is an aldehyde, therefore you have different physical and chemical properties. Okay, and the most important that I want you to more or less memorize the structure. So this is the what we call D-glucose. So that's why you call it dextrose. So the D comes from the name dextroglucose, because the other enantiomer of glucose, so if you have the mirror image of this structure, this will be L-glucose. So levo glucose. Well, basically, we cannot utilize it for energy because our enzymes, our body, biochemically, the L-glucose cannot be utilized by our body. So it's not natural to have an L-glucose in the body. It can be made in the laboratory. It's actually sweet. So, but the problem with the L-glucose is it's expensive to make in the laboratory. And since our body cannot consume it, theoretically it can be an artificial sweetener. So our body will just, you can use it for its sweetness, but it will just go through our body without being digested. So this is the structure of the glucose. So you can more or less remember the structure of the glucose. You remember your four fingers of your right hand. You place it here. Okay, you have your fingers here of your right hand. So if you raise your middle finger, the OH will be the one on this side, on the left side. So that's an easy way to remember the structure. So you have a C double bond O on the top and the CH2OH at the bottom. Okay. So it's found in fruits, vegetables, corn syrup, honey. Sucrose is made up of glucose and fructose. In lactose, glucose is combined with another sugar called galactose. In maltose, it is two glucose units combined with each other. Also, it makes up polysaccharides. So glucose units make up starch, and as mentioned, that's for plants. Glycogen, we store excess glucose as glycogen. And interestingly, the composition of your paper, it's actually made up of cellulose, mostly cellulose. Cotton is made up of cellulose. So you might be asking, if it's made up of glucose, why can't we eat paper? Okay, so that's one of the things you have to think about while we discuss this topic. Okay, we can digest starch and glycogen since it's made up of glucose, but not cellulose. So the D-glucose or dextrose that we use in the hospital. So the normal blood glucose levels should be 70 to 110 milligrams per deciliter. So sometimes our blood glucose level fluctuates, especially after we eat. So it can go beyond that. So in order to get the normal or the the normal blood glucose levels, they ask you to fast for 8 hours before they get your blood. Okay, so that will get you more or less, that will be a stable level of the blood glucose level you have in your blood. So excess glucose, once you eat a full meal within the day, that will be stored as glycogen in animals and humans. In plants, they convert the energy from the sun to create glucose as well, but they store it as starch. In our case, we have to eat plants in order to get the glucose, or we can get it from other sources as well. So the glucose source comes from mainly plant material. So it's within the day can be stored as glycogen, but if you eat too much, the excess glucose will eventually be converted to fat. Okay, so that's true, especially when you like to eat and then you you have late night snacks and then you sleep. Since you're not utilizing it for energy, that will be converted to fat. So it's not a healthy habit to have midnight snacks. Okay, we have mentioned this as hormone before. So insulin is important in regulating blood glucose levels. So this is done by stimulating the uptake of glucose in tissues and the formation of glycogen. So it's very important uh, in the maintenance of the blood glucose levels. So if you cannot create enough insulin, if your body does not create insulin, then you have this disease called diabetes. Okay, so if you have insufficient insulin, uh, sometimes you may be hypoglycemic, meaning the blood glucose level goes below 70 milligrams per deciliter. So that means you can become weak or even faint, lose consciousness because of the lack of sugar, blood sugar in your circulatory system. So on the other extreme, you may eat too much carbohydrates and sugars. So the glucose will not be metabolized, and this may accumulate in your organs. So your eyes may form cataracts. Eventually, it might damage the kidneys. So that's the only you. This is the way the diabetic patients die when they do not monitor their diet. They eat too much sweet, so this sugar um, accumulates in their organs, and this will lead to the damage of their organs, and eventually they can die from that. So as long as you monitor your diet, if you're diabetic and you monitor your diet, you won't be uh, prone to organ damage. So as long as you monitor your diet, or more expensively, you inject insulin. So insulin is expensive. That's why if you don't have the financial capability, the best way is to monitor your diet. Sometimes you have to have a readily available glucose sugar source like cracker crackers or something to eat nearby when you become hypoglycemic. Okay, another important monosaccharide is fructose. So it's the sweetest of the carbohydrates. So notice that the C double bond O is found in the second carbon. So it's the sweetest of all the carbohydrates. It's found in fruits, fruit juices, and honey. So in our bloodstream, we have an enzyme that converts glucose into uh, the fructose, or vice versa, fructose to glucose. Inside our cell, we when we convert glucose, eventually we utilize it for energy. The glucose structure is converted to fructose until it is broken down into two molecules. We will discuss that later in metabolism. So fructose is bonded to glucose in sucrose, which we know as the common table sugar. So sucrose is a disaccharide. So the level of sweetness in naturally occurring carbohydrates, fructose is the highest, 1.75. Um, relative sweetness scale. Here you have artificial sweeteners. You have sorbitol, aspartame, saccharin, and sucralose. So these are sugar substitutes. Okay, another structure, another monosaccharide is galactose. So you have the fourth carbon, the OH group is left on the left side. So this cannot be found free in the free form in nature. It exists in the cellular membranes of the brain and nervous systems. Okay, so lactose is a combination of galactose and glucose. So when someone has a disease called galactosemia, it means they lack the enzyme to convert galactose to glucose. So what is the consequence of that? Since galactose is not metabolized, then it becomes accumulated in your body. So it becomes accumulated in the blood and tissues. So in the brain, it might lead to mental retardation. In the eyes, it will lead to cataracts. So they need to stay away from dairy products or anything that has milk in them. Okay, so uh, this is hereditary since your DNA doesn't have the code to make the correct enzyme that converts galactose to glucose. Okay, going back to chirality. So where do the terms D and L glucose come from? So we go back to the stereoisomers discussion, stereochemistry discussion. So all carbohydrates have one or more chirality centers or stereogenic centers. So you have a chiral carbon here for your glyceraldehyde. So if the position of the OH is on the right, we call it a D-glyceraldehyde. If the OH group is found on the L, it is the L-glyceraldehyde. So again, all the carbohydrates that are naturally occurring and that we can metabolize are in the D form. So when we're talking about the where does the name come from, the D glucose comes from the positioning of the wait so the position on the fifth carbon, the OH is found on the right. So that's D glucose. If you have a mirror image, the OH is found on the left. So that's L glucose. Again, the Fisher projection is a faster way to draw a tetrahedral structure. So if you remember, when you draw a wedge, it means it's going towards us. If you're going a dashed line, these groups are going away from us. So think of this as the tetrahedral structure, as a tetrahedral structure. So when you convert it to a Fisher projection, you can more or less visualize the tetrahedral structure. So horizontal lines represent bonds projecting forward from the stereocenter, while vertical lines represent bonds projecting to the rear. Okay, so only the stereocenter is in the plane. Okay, so that's why we call this the D-glucose, because of the positioning of the OH group on the right side. If you have a mirror image, this will be the OH found on the left. Okay, so that's where it gets its name. Okay, another thing. We aldehydes and ketones, since they have a carbonyl group, they have a tendency to react with alcohols to form what you call hemiacetals. So the RO group here may attach itself to the carbon. Okay, so I'll just annotate this one. So this group, the ROH, so the oxygen will attach itself to the carbon, leading to this oxygen losing one bond, and then eventually it will have a bond with the H. So you have the OH here on top, and the OR from the alcohol That's here. So this is a hemiacetal. This structure is unstable. Okay, but it will be more stable if both groups are part of the same structure. Okay, so cyclic hemiacetals form readily when the hydroxyl or carbonyl groups are part of the same molecule. So you can twist this bond around to form something like this one. So you can twist it around and you form this. So this is the carbon. Oops. Then you have another group here which contains the OH. So I twist this bond around so that the OH is found here. Okay, so I'm just showing you that I twisted around the bond. So eventually, I can position the oxygen very near to the carbon, so it will form this cyclic hemiacetal. Anyway, we will continue with the discussion. I'll just give you a video uh, to watch first before I discuss more in detail.